Ladies and gentlemen, welcome to the 2013 Royal Society GlaxoSmithKline Prize Lecture. I'm Jean Thomas, I'm the Biological Secretary, and I have the housekeeping duties of asking you to turn off your mobile phones, please, because the lecture is being recorded and webcast, and also to tell you that in the, I hope, unlikely event that there's a fire, you don't go out through the usual doors, but you go, because of the snow and whatever, you go out through these doors instead. So 2013 is the Royal Society's Year of Science and Industry. And this is the year when the Society will showcase excellence in UK industrial science and strengthen links between the Society, industry and academia. The Royal Society recognises that world-class research and development in the UK industry is essential for transforming innovative ideas into commercially successful products, into economic growth and securing the science base. And it will be proactive in anticipating, understanding, and responding to the needs of industry scientists. Symposia and meetings with high industry interest have been added already to the Society's calendar, which already includes long-standing initiatives in scientific excellence, such as the Royal Society Industry Fellowship and the Brian Mercer Awards for Innovation and Feasibility. So the Year of Science and Industry will bring a renewed focus on engaging with the industrial sector to develop cogent arguments that high-level investment in the UK science base is essential for international competitiveness, something we would all, I'm sure, sign up to. Now to the prize and the lecturer. The Royal Society GlaxoSmithKline Prize and Lecture is awarded biennially for original contributions to medical and veterinary sciences published within 10 years of the date of the award. The prize consists of a very nice gold medal, even nicer cheque for £2,500, and the recipient is called upon to deliver an evening lecture at the Royal Society, which is why we're all here this evening. And this is really a capacity audience, and the reason we're a few minutes late starting is that there is an overflow room. And I don't remember that certainly in the last 4 years of chairing these evening lectures. So Adrian's really pulled in a crowd tonight, so no pressure there, Adrian, at all. The award was initially established following a donation from the Wellcome Foundation. First award was made in 1980, the centenary year of the Wellcome Foundation, and since 2002, it has been supported by GlaxoSmithKline Limited. So this year's recipient of the prize is Adrian Bird, an old friend and colleague, and I'm delighted that he's received this award. Adrian has held the Buchanan Chair of Genetics at the University of Edinburgh since 1990, and he's a member of the Wellcome Trust Centre for Cell Biology in Edinburgh. His research focuses on the basic biology and biomedical significance of DNA methylation and other epigenetic processes. His laboratory identified CpG islands as gene markers in the vertebrate genome, and he discovered proteins that read the DNA methylation signal to influence chromatin structure. Mutations in one of these proteins, MECP2, and I'm sure we'll hear a lot more about this this evening, causes the severe neurological disorder Rett syndrome, which is the commonest genetic cause of mental retardation in females. Adrian was made a Fellow of the Royal Society back in 1989. He's received several awards, numerous awards for his work, including, notably, the Louis Jontet Prize for Medicine, the Charles Leopold Mayer Prize of the French Academy, and the Gairdner Prize. This evening, it's the turn of GSK and the Royal Society to give him this special Royal Society GSK Prize, and he has to give his lecture in order to earn that, and his lecture is entitled, as you can see, Genetics, Epigenetics and Disease. So, Adrian, over to you.
Thank you very much, Jean. Thank you very much for this award. It's a great honor to be asked to give this lecture. And thank you very much for braving the elements to come and listen. I think probably the title of Genetics, Epigenetics, and Disease is broad enough that it sounds like it's going to change all our lives in this next 45 minutes. But in fact, I'm going to focus on a relatively small part of it ultimately, but I'm going to start off reasonably broad. There's one deliberate mistake on the first slide. I hope it's the last one. It's the year. So let's go back in time to the draft sequence of the human genome, because this was heralded as a time when biology really became a hard science. If you like, it was seen as the beginning of the end. We now knew the entire code of we knew the sequence of all the genes required to make a human being. But it's pretty clear that it was actually the end of the beginning, and the somewhat apocalyptic predictions that now one simply had to automate the discovery of all the medical innovations that would result from the genome sequence was premature. In fact, it's likely, in my opinion, that there's still another century of biology to be done, and this will be an exciting century of discovery, converting the promise of the genome into the reality of biomedical applications. And one of the issues, I think, that we would really love to be able to solve, a big question, if you like, is where DNA, despite being the thread of life, you can put it in a tube and gaze at it for as long as you want, and it remains utterly dead. So the question is, really, what does it take to make it alive? When Craig Venter synthesized a bacterial genome, an important synthetic biology milestone, it had to be put into a living cell before it became alive. How can one bypass that? As the chemists say, you only really understand something if you can make it. We can't actually make life, but it would be good to know some of the rules required to do that. So some key unanswered questions about the genome that remain, and this is only a selection. First of all, a basic fact: genes make proteins. Here is the chromosome. Here is the sequence of the genes. There's the RNA. It encodes the sequence of the amino acids that lead to the protein that folds up to then do all the lifelike things that are required. But how are only the right genes expressed in a cell type? This has been a question, a long-standing question. Do we know the answer to it? Why globin is expressed in blood cells and keratin is expressed in skin cells, etc. We approximate knowledge of it, but actually there's an enormous amount to find out. Most of the genome is actually inaccessible. If this is this gray, it's rather difficult to look at this picture, I think, because the DNA is gray and it looks as though it should be in the background, but this is a nucleosome, the repeating unit of the chromosome, if you like, the fundamental repeating unit, and the DNA clings to the outside of it, and proteins that want to make genes active can't actually get at the DNA properly. So how does the gene activation machinery gain, and how does it keep access? Again, we have some beginning answers to this, but we don't by any means have a full picture. Protein coding DNA sequences are only 1% of our genome. So if you look at a piece of the human genome, you see these vertical stripes correspond to the bits of this gene that are separated from each other. In fact, genes are fragmented, and they are a tiny minority of all the DNA. What is the rest of it for? There's a vast majority that we can't explain. This isn't the case with all organisms. This, for example, is yeast. And you can see now the genes are packed together. It's difficult. It used to be said casually that the rest of this DNA was just junk. But now it's sort of almost politically incorrect to call it junk. It's particularly after the ENCODE project, which found lots of potential regulatory sequences throughout here. So this other DNA is doing stuff, and perhaps it's doing stuff that makes, for example, humans and other mammals far more complex than yeast. So finally, there are questions, almost sociological questions. Does the environment have any impact on gene expression? And this is a question I'll allude to in a moment, but it's not one that is the main subject of this talk. So I put in the title Epigenetics because our work is quite often described as epigenetics. It literally means above or in addition to genetics. But the definition has been controversial, and I'm just going to skim somewhat lightheartedly over some of this because it's at meetings to do with epigenetics. One can see various opinions expressed with varying degrees. This one, I believe, was in Barcelona with great vehemence. So let me just try to sort of consolidate this. The original epigenetics definition comes from Conrad Waddington, who was actually my predecessor as Buchanan chair of genetics in Edinburgh. And what he meant was in contrast to preformationism, that the development proceeded by the gradual unfolding of the information in the genes to produce the whole organism. So for him, how information of the genes is read during embryonic development to give the whole organism was the essence of what epigenetics was about. We would now call this developmental biology, how the genotype gives rise to the phenotype. But it's acquired a sort of a special status in epigenetics really because of this iconic picture, the epigenetic landscape. I'm not going to dwell on this either because, quite honestly, having had it explained to me several times, I'm never totally sure exactly how this helps. It's a picture of a ball rolling down a hill. The number of options for the ball get progressively less. But I don't feel that this encapsulates anything very useful. This, however, is a fundamentally important question that remains on our agenda. Second, a definition of epigenetics, which is rather different, has actually different origins, epistemological origins, how characteristics are inherited across cell or organism generations without changes in the DNA sequence itself. An example of this is this cat, the so-called tortoiseshell cat or calico cat in the US, which has these patches of fur. It has two X chromosomes. One of them has a gene that gives black fur, the other one has a gene that gives orange fur, and cells early in development inactivate one or the other of those chromosomes for reasons we don't, which I will come back to actually a little bit later. And you get a patch of skin because the cell that originally inactivated the orange fur gene gave rise when it divided to cells that did exactly the same thing. So that was inherited. All the gene, the DNA is still there in these cells, in the orange ones and the black ones, but there is a difference that is inherited, and that's epigenetic according to this definition. So heritable traits of this kind might be influenced by the environment, and this is sort of revitalized an ancient argument about nature versus nurture, where nature is genetics, the idea that where our genes are in control, and nurture is the opposite, the idea that our environment determines who we are. Of course, it's a mixture of both, but epigenetics has given a new lease of life to the nurture argument, and so one can see articles such as this, and there are many examples I could have chosen, why your DNA isn't your destiny. The new science of epigenetics reveals how choices you make can change your genes and those of your kids. Now, I'm not an expert on some of the epidemiology behind this, but the molecular biology, in my opinion, is far less convincing than it is for other aspects of epigenetics. It is, however, an extremely interesting idea that the environment can give rise to changes that get passed on, but it is systematically overstated in a lot of places one finds it described. So one has to be circumspect about this kind of argument, in my opinion. There are a couple of excellent examples in plants, in worms, where the immunity is involved, but some of the more sociological aspects, in my opinion, require further evidence. So I'm sticking with this as my example of heritable epigenetics. It's closer to the molecular biology we actually understand. So epigenetics 3, biological significance of the epigenome. Another definition, it's arisen pragmatically. What is the epigenome? Well, here's a genome of a cell. It's the chromosomes that were obviously designed for an experiment because there are fluorescent pinpoints here. Ignore those. That's a human chromosome complement. If you explode those chromosomes, you see beads on a string. And this is that repeating unit I referred to earlier, the nucleosome, with the DNA going around the outside. It looks like beads on a string. So the epigenome refers to markings of those beads, of that string of beads, in such a way that it is regionally adapted to its function. So, for example, there can be a region where a gene is stably on, and there is a whole plethora of marks that appear that reinforce that decision. And similarly, stably here, a gene off, such as the black coat gene in an orange patch of fur. And again, you get adaptation. And this is the epigenome, and the study of what that epigenome means is another definition of epigenetics. So you have DNA methylation here, where these methyl groups are added to the DNA. You can't do much to DNA without changing its important properties. Almost the only thing it seems you can do is put these methyl groups on, and even that is bad in a way I don't have time to go into. It causes an increase in the frequency of mutations. But by far the most elaborate way of marking the chromosomes is via these beads, which invisibly on any of the structures I've shown you previously have tails. And these tails are basically ticketing entities that you can add chemical information to, that the cells can write information in the form of chemical alterations. And so you add this that says stably on or stably off. Again, we have the broad outlines. We can correlate quite a lot of these with activity and silence. But if you were to ask exactly what each of these modifications does, we still have a lot to learn. So, if you like, epigenetics is the structural adaptation of chromosomal regions so as to register, signal, or perpetuate altered activity states. And importantly, proteins that read these marks, write the marks, or erase the marks, remove the marks, are implicated in human disease. And quite a lot of excitement in pharma, including GSK, is devoted to finding out what these drugs might be good for in terms of human disease. So epigenetics then embraces key unsolved problems in biology. How the genotype gives rise to the phenotype, that's the Waddington one, how traits are inherited across cell or organism generations without changes in the DNA sequence, on how structural adaptation of the genome facilitates gene activity programs. As far as I'm concerned, this is not a word one needs to dwell on with sort of almost a theological interest about what it means. Everything it possibly means is interesting, so let's get on with studying it. And I like to think of it as how the genome is organized and managed to make DNA, if you like, come alive. So CG is one such signal. It's one of those marks. And you'll notice CG is not actually a mark, it's actually a sequence. It's a 2 base pair sequence. DNA sequences that recognize proteins are usually longer than that because they're rarer. If you have a sequence of 1 base, every few bases you'd come across it and it doesn't have much information. 2 bases is not much better. But nevertheless, as you'll see, CG is used as a genetic signal and also as an epigenetic signal. So here's a piece of DNA flattened out so it's no longer helical. The 2 strands are anti-parallel, and CG is paired with itself. So CG pairs with CG. This TA is paired with itself, but it's not nearly so interesting. And one of the things we'll talk about that can happen to CG is that the C gets methylated. And that since there are 2 of them, that can be a symmetrical event. And it looks like this. They sit in the major grooves. I've already shown you a different picture, though with less vulgar coloring, that shows the 2 methyl groups sitting in the major groove. And they influence interactions between proteins and DNA. So what are the features that adapt CG for a genome signaling function? The first is that, as I've mentioned, you can get it in 3 different chemical forms. Actually, there are more than 3. There are another 2, but it's not yet clear whether these are biologically important or just byproducts. At least it's not clear to me. You have CG unadorned, you have CG methylated, and you have CG where the methyl group has had an oxygen added to it and it becomes hydroxymethylated. So it exists in different forms. Specific proteins are attracted or repelled by different modified forms. And we're going to talk more about that. Highly variable in frequency. So the frequency of CG, despite the fact that it's just a 2 base pair sequence, is dramatically different as you're going along the genome. In the bulk of the genome, 99%, it's quite far apart. These lollipops represent CGs. The lollipops that are solid represent methylated ones, and the open ones represent unmethylated ones. So 99% of the genome has not many CGs, and most of them are methylated. But then there are these clusters where the density is about 10 times higher, and these are the so-called CG islands. They are interesting because they sit right on top of the control regions for genes. So here's a gene, it's red in this direction, and then these blue bits are spliced together to make the messenger RNA. And sitting right on top of the promoter is this CG island. And this amounts to about 1% of the genome. There's another one there. And here's a biological consequence of the methylation. If you look at this CG island, it can, under certain circumstances, this happens on the inactive X, this happens at imprinted genes, it happens at germline genes in the soma, it happens in cancer aberrantly, it gets methylated. And when that happens, you shut down transcription of the gene. And because methylation, something I haven't gone into, is relatively stable, it can be transmitted from one generation to another, if you like, copied when cells divide, one cell generation to another, this is quite a stable silencing. So one of the things DNA methylation does is it shuts down the expression of genes. So we're going to talk about specific proteins that are attracted or repelled by modified forms of CG. And I'm going to start just with a protein that recognizes unmodified CG. So it can't recognize this or this. So CFP1, sorry about acronyms, it's a protein that recognizes, it was discovered in fact by David Skolnick. It binds to non-methylated CG. I don't know why I've drawn the DNA at this jaunty angle, but it just meant to show that it's interacting with it. And it also interacts with a complex of proteins, an enormous complex, well, relatively big complex, called SET1. And this complex does something to the nucleosome. We've seen this before. This is the bead on the string, the DNA going around the outside. When you determine the structure of something like this, you don't find the tails, the things that you write on. And so I've drawn them freehand. Nobody actually knows where they are because they're so floppy they don't come up in the X-ray structure. But amino acid lysine number 4 gets methylated. And this is done by this complex. So we have a protein that binds to non-methylated CG that recruits a complex that methylates this. Now, why is that interesting? This is a mark of active genes. So if we look where CG islands are, or CPG islands as they're more often called, in fact, here are the CPG islands. I'm not going to tell you how we know they're there, but you'll notice this gene is going this way. There's a CPG island at the start of it. This gene's going this way, actually bidirectionally. There's one going this way, one going this way. There's the CPG island. So there are all the CPG islands. There's the RNA polymerase, the protein, the machine that makes, that starts genes to be copied into messenger RNA. And it's just at the beginning of them because this is a particular form of RNA polymerase that is only at the beginning of genes. And here is this mark, H3K4ME3, which means this purple blob on this tail, which is put on here. So we have the non-methylated CG cluster here, and we have the mark. And this mark is involved in gene expression. So could it be that the proteins attracted by the CG brings in this, and that's what causes this mark? If you look, as we did, where the protein is, it coincides with the CPG islands. So it's in the right place. If you take it away, the K4 trimethylation, these peaks here go down, and that's consistent with the idea that this is reading the CPG island signal. But the key experiment is really that Pete Skeen and John Thompson did, is to insert a piece of CPG-rich, CG-rich junk into the genome, real junk. In fact, it's not actually quite junk. It's a jellyfish gene that's been optimized for expression in humans, lacking any control sequences, just insert it into the genome. So you make a CG island-like sequence with a cluster of CGs. Now, are you creating a new H3K4 trimethylation peak? So here's a map of all the CGs. The vertical lines show where they are. And this is what we've inserted. and you can see the density of CGs has gone up. And now you can plot that density. Now, where's CFP1, the protein that binds CG? There it is. We've now got a new peak of it. And what about H3K4 trimethylation? It's there too. And you notice where there's most CG, there's most of that modification. Have we just made a gene? In other words, all the stuff that does gene-y things is there? No, because there's no RNA polymerase there. So this is just the DNA sequence talking to the chromatin. One can do this with other sequences and verify that it's the case. So a CG-rich piece of DNA creates a new region of H3K4 trimethylation, this active promoter mark, even when there's no active promoter there. So the presence influences of the CGs influences the chromatin structure via this link between the DNA binding protein and the set enzyme complex. And other CG binding proteins also recruit modifying enzymes. In fact, for a long time we were used to the fact that CG islands existed, but we didn't really know what they were for. And actually one almost forgot to ask, well, they're always there, what are they for? In fact, it now seems very likely that they are platforms to set up appropriate genome structures at gene promoters. Very important function. And there are other proteins that bind CG that recruit other things to them, and this is a rapidly growing area. So suddenly we find that the CPG island is a structure of biological importance, and we're starting to disentangle how. So here's a CXXC protein, which has this domain. CXXC is the name of the protein domain that binds CG. It comes in wearing this ludicrous wig and creates a sunny promoter, gene activity friendly region in the genome. If there's a methyl CG, it comes in and it goes away. It can't bind. So now I'm going to turn to, for the rest of my talk, proteins that bind to the methyl CG mark, the one with the purple blobs on, the purple blobs that were on the DNA, not the purple blobs that was on the histone tail. So what binds this form of CG? Well, a protein that we found a long time ago is MECP2, and this binds specifically, and I'm going to show you some of the prehistory of this protein. First of all, a picture from the paper, and you could either take from this how prescient he was to be able, in 1992, to find this protein that turns out to be so interesting, or you can think he's been working on that protein for 21 years and he's still not quite sure where it does. You can take your pick at the end of the talk. So this shows how we first found it. We run the proteins that are in a nucleus on a gel, and then we probe them with a piece of DNA that's labeled and methylated, and then the same sequence of DNA with no methylation. And clearly there's a protein that binds when it's methylated and at about 84 kilodaltons and doesn't bind when it's not. And we now know a structure for this in atomic detail. Here are the two methyl groups sitting in the major groove, and this is the domain of this protein that interacts with them. So we were happily studying this for blue skies reasons, to try to find out a protein that read DNA methylation and therefore a reader of DNA methylation and find out what it did, when Huda-Zoghbi showed that the gene that causes Rett syndrome, an autism spectrum disorder, is almost exclusively, more than 90%, MECP2. So this is the gene that is mutated in Rett syndrome. So what is Rett syndrome? This is a film just taken from YouTube, not somebody I've ever met, but you can see the characteristic features of Rett syndrome, which involve this repeated hand clasping and a period of apparently normal development for 6 to 18 months, and then regression, progressive encephalopathy, repetitive hand movements, breathing arrhythmia, a profound problem, but nevertheless a life expectancy of about 40 years on average. So there is no effective treatment and 24-hour nursing is required. So this was all caused not by a brain gene that was what was being looked for by everybody. Those were in the days where you thought the gene would have something to do with the tissue that was affected, but a basic housekeeping protein that reads DNA methylation that's expressed in every cell type. So why does this disorder only affect girls? Well, you probably guessed it's because it's on the X chromosome. Males are always more affected by mutations in genes on the X chromosome than females because they only have one X and females have another one, which can compensate. And males die. There is no male Rett syndrome simply because males don't survive. So then you have a new mutation. Nearly always as it happens, like many of these things, paternally derived. And then proceeds X chromosome inactivation. I remember, so in order that females have only the same number of functional X chromosomes as males, they shut one off. And this happens in random cells, and I've shown you the example of the cat, and I'll show you the cat again. This gene, this cell inactivates, or this progeny of this cell inactivates this X chromosome, this one inactivates the other one, and this then is inherited. So this is the epigenetic inheritance phenomenon. It's passed on. And the end result is, there's the wretched cat again with his different things. I will show you a different example of that in a moment. But the point to be made here is that the brain and, in fact, the other tissues of a Rett patient consist of a mosaic of a salt and pepper mixture of cells that are functionally normal with respect to MECP2 and cells that are functionally without MECP2. So here, for example, is the phenomenon. A new mutation arises. Sometimes that mutation is the only MECP2 in the cell, and the other time it's invisible and you get the wild type expressed. So this is the mosaicism. Now, the equivalent of the cat picture in the brain, though, is rather different. This shows the dentate gyrus, which is a region of the hippocampus, which is part of the brain, from a mouse. I haven't talked about the mouse yet in any detail, but just to show you that you can see patches. It's probably better to see it here in the merge. This is MECP2, and it's in blobs, and there are gaps. But actually, there aren't gaps in the nuclei staining. And so there are patches of cells here that inactivated the functional MECP2 gene, and there are other patches here that inactivated the non-functional MECP2 gene. And you see these patches. The point I'm making here is the patches in a cat are gigantic and involve millions and millions and millions of cells. The patches in the brain are, for reasons we don't quite understand, tiny. And so you get a big admixture of functional and non-functional cells in this tissue. So the first thing we did when we found this out was to make a mouse. We were going to make a mouse anyway for our blue skies reasons, but now we were energized, I would say, and that energizing has continued to the present day by the fact that we were working on a human disorder and were actually in touch with a community of people who are affected by it. So if you take a normal mouse, it lives in this green state for a long time. But the MECP2 minus mouse, the male, the equivalent of the human male that doesn't survive. And these colors are meant to indicate that they get symptoms, get worse and worse, and eventually die. The female, and this shows a sign of neurological symptoms in a mouse, it does this hind limb clasping, and it does that at this blue stage here. Initially, there is no observable phenotype, but later on, they become ill and subsequently die. The females, and these are really the true model of Rett syndrome because they're heterozygous, as geneticists say for these, this mutation, they're fine, and that's how you keep the line going. They breed for several months. And a mouse at 6 months of age is quite an old mouse. It's had quite a few litters. But then they suddenly hit a wall, and they become immobile, and they develop all the other sorts of symptoms, including hind limb clasping, arrhythmic breathing, lack of mobility, that characterize the Rett-like phenotype. And there's a dramatic change in their behavior, but it's stable, just as it is with humans. So if you like, the MECP2 deficient mouse is actually quite a good model. Not all models are particularly persuasive, but it's quite easy to persuade skeptics that this is a good model of this disorder, because a lot of the things that MECP2 seems to do in humans, it also does in mice. So we're armed with this model. Now, how are we going to find out what MECP2 actually does and how that's connected to the function of the brain? Because that's what's gone wrong in Rett syndrome. Well, the big resource you always have is in genetic disorders is the mutations that give rise to the disorder, particularly if, like Rett syndrome, they're all new mutations. This does not run in families. The males don't survive, and the females don't reproduce either. So it doesn't run in families. Everything's a new mutation. And so this is the sort of picture you get everywhere, absolutely all over the place. But I will point out to you that these frame shifts, these gray ones, the longest bars, everything downstream of that is disrupted because the protein goes out of frame and you start making junk afterwards. So they don't mark the spot where there's an important bit of this protein. They only tell you that this is the boundary between, and everything downstream gets lost. The other ones, the nonsense mutations, also stop the protein. That's why you put X here. They just terminate the protein. The ones that are most informative are the missense mutations, because what's happened there is you've put an alien amino acid, one single subunit of the protein, in the wrong, is wrong. Everything before it is fine, everything after it is fine, just that one amino acid is wrong. And so this is telling you the really important bits. And if you notice, the blue ones, which are the missense, are not randomly distributed. So we went into the database. Now, of course, for a lot of disorders that look like they might be related to MECP2, and there's more than Rett syndrome, I don't have time to go into that, people look at the database and they they sequence. And so there's an awful lot of polymorphisms, a lot of changes that are not associated with disease. The one way of being sure it's associated with disease is to look for mutations that are not found in the parents. They're only found in the offspring, because then the probability that that is a functionless genetic variant is vanishingly low. And you see very specific domains here. Interestingly, and I don't have time to go into this, there are now more and more exome sequences. People are sequencing genes of normal individuals or for people who have other things. And so you can find all the missense mutations where there's no obvious effect. And what you notice is that this cluster here, for example, doesn't have any genes with no obvious effect. This cluster here, the same. So you can use the normal polymorphisms as a way of seeing the inverse of what you see with the mutations. So now we have two domains. What's this domain? Well, I've labeled it MBD. Actually, what that stands for is methylated DNA binding domain. I showed you the X-ray structure of that bound to methylated DNA. That's the bit that contacts DNA and brings this in. And many of these mutations prevent that. So we're pretty clear what's going on. I'm just going to tell you a couple of things about that domain. The first thing that emerged when we studied it was you tend to think when you find a DNA binding protein that it goes to specific targets, and then it does stuff there, and those targets are its main function. But actually, it turns out that MECP2 is incredibly abundant, specifically in neurons. And in fact, there are 17 million molecules per nucleus in a cell. This is a lot. It's 1 every 400 base pairs. It means there's enough to coat the genome, and that's actually what it does. DNA methylation goes up and down along a chromosome, so this is a very low-resolution picture, and MECP2 goes up and down in exactly the same way. So it's not in special places. It's all over the place with somewhat different densities, and it's very, very abundant. It doesn't behave like a transcription factor, which goes to specific target genes. It binds globally. So that's that domain. Let's now talk about this domain, and this is was more interesting to us because we had no idea what it might be. So hypothesis was that this region binds to DNA, and then this region binds to some sort of partner that it brings in, and that's its job. And you can't mutate that because it fails to do that. And Matt List really led this aspect of the project. What we did was we made a mouse with a green fluorescent protein tag on the MECP2, and then we pulled down that tag from the brain, an extract of the brain of the mice that had it, and then we asked what came down with it. Those are the partners. And by mass spectrometry, we found these proteins. This is the list of the top 8. Interestingly, well, MECP2 came down. That's a relief. You expect, if it didn't, you'd have a real problem. Then 2 proteins that transport it into the nucleus, but then these 5 subunits and more acronyms, I'm afraid, of a complex that's well known. This is a huge complex, more than 1 million Daltons complex, which contains a histone deacetylase 3. So what a histone deacetylases do is they remove a mark on one of the tails. That mark is associated with activity. If you remove that mark, you work against gene activity. In fact, you silence gene expression. So this is a complex that reinforces the silence of gene expression, shuts genes down by removing this methyl group. So here it is. There's the methyl group. PowerPoint extravaganza goes. So this is, it's well known to bind nuclear receptors, and it also now, we find that it binds to MECP2. Now, where does it bind MECP2? Well, it binds it, you won't be surprised to hear, exactly in this second domain. And all of those mutations that cause Rett syndrome in this second domain abolish the interaction with this complex. So this mutant protein can't bind DNA. This mutant protein can't bind NCOR SMART, which is the unfortunate name for this complex. And also, you lose the ability to shut down transcription. So we now have this fairly persuasive model, I think, that MECP2 is a bridge. It's a bridge between DNA, there's a methyl group, MECP2 is attached to it, it's brought in this complex, which is a gene silencing complex, and if you have mutations in the DNA binding domain, it can't bring it in, and if you have mutations in the complex interaction, you can't bring it in either. So MECP2 then, and other proteins that bind methyl CG, and there are others about which we know something, they come in, bind, and instead of creating the sunny atmosphere, they create a foggy, transcription hostile environment. I was going to say like Edinburgh in January, but it's not really transcription hostile in Edinburgh. We express our genes perfectly well. So then the big question is, have we got any further now? We know it's likely to be a repressor, and to you, it probably seemed likely that was always going to be the case. DNA methylation represses transcription. This binds DNA methylation. What more natural than it represses? Actually, it's a very controversial area as to whether or not it does repress transcription, and I feel this is an important advance. So the question is, what transcription does it repress? Because it's not obvious when you look in the brains of these mice. Histone acetylation up, histone H1 is up, the epigenome is disorganized, expression of some genes is up, other genes is down, other genes are down, and some are unchanged. And these effects aren't very big. So very, very briefly, I'm going to say you could be controlling the activity of specific genes, you could be controlling transcription in response to neuronal activity. Only when neurons fire, something happens. This protein actually gets phosphate groups added to it. Maybe that's something to do with it. Or dampening of transcriptional noise. And this is a boring sounding possibility. It just kind of sits on the genome and keeps everything down. But there's some evidence for that. We know that the transposons, which are selfish elements in the genome that like to jump around, when there is normally, they jump around represented by these yellow dots, a very small amount. When you don't have MECP2, they jump around an awful lot more. And so, in other words, MECP2 is preventing the expression of the RNA that allows these things to move. And this is no function for the organism. It's actually something it would prefer to keep quiet. So that's noise dampening. So this question is unresolved. So now in the last part of my talk, I've left you there with, that's as far as we've got with the molecular biology, I'm afraid. But I think we're now making progress. Now we know we have this bridge model. Let's now talk about the pathology and trying to get all the way from the molecular biology up to the patients and our surrogate for the patients, which is the mouse model. So what we really want to do is have a molecular description of the lesion in MECP2 and account for all the steps to the brain of the patient so that we can understand. And this requires we know an awful lot more than we do now, for example, how brains work. So we're trying to bridge this gap. Now, our involvement in this is really to do with one specific question, and that is this one: can the symptoms be reversed? In the pathology, as observed in post-mortem brains and as seen in the mouse, is that neurons are slightly simpler. So if you put a bullseye over the center of a neuron, then its arms are more complex and branchy in the normal animal compared to what they are in an animal that doesn't have MECP2. That's about it for pathology. There's no cell death, so it's not a neurodegenerative disorder. It's not like Parkinson's or Alzheimer's or Huntington's where nerve cells die. It's just a kind of shrinkage. They become underpowered neurons. So the question then arises, if they're not dead, if we put MECP2 back, can it be reversed? And I'm going to tell you about that and then our attempts to do some therapy based on that. So how do you do this experiment? Well, what you do is you take the MECP2 gene, you put a stop in it, which is just a chunk of DNA that is poisonous for transcription, and then you flank it with sequences that mean that when you want to, so that then causes transcription to stop. You let the animal grow up, it has no MECP2, and it becomes ill as a result. And then at your chosen moment, you remove that stop and start transcription again. And you can do that in ways that have been published and I can tell you about if you want to know afterwards. But this works. That's the first surprise. It actually works. And the reason why it works is because Jackie Guy, who's an unbelievably talented person in the lab, took charge of these experiments. So I've shown you the mouse that's wild type, the mouse that's male. What we're going to do now is look at a mouse where it's male, it's in the death zone, if you like, and we inject it with tamoxifen, which is the way we trigger the deletion of the stop cassette. Does it work? Well, this is MECP2 in a normal mouse brain. This is in a stop mouse brain, so the stop works. This looks like it's cells that haven't been stopped, but actually it's blood cells that autofluoresce. And then you treat with tamoxifen, and back comes the MECP2. So that works. And then here's a mouse on the day we started the experiment. So it had grown up with no MECP2. It had the classic symptoms of the MECP2 null mouse. It has this tremor. It has arrhythmic breathing, which you may be able to see in the flanks. They breathe, and then it stops, and it breathes, and it stops. And it doesn't move. And the film's much longer than this, and it still doesn't move. And then when you humanely suspend it by the tail, it does this hind limb clasping. So then the question is, what does tamoxifen do for that? And then this is the same mouse a month later. Under our animal license, this mouse would not be able to survive for more than a week or two at the most. And here it is a month later, remarkably healthy, and it went on to live, I won't say a natural life, but quite a long life. So this is an unexpected finding. We didn't expect it, and turns out nobody else did either. And for that reason, it's turned out to be quite important in the field. This somewhat unedifying image of a mouse, I will leave you with, and go on to the females, because those mice are young. They're only 6 to 8 weeks old, and so it could be that they're young and plastic, and reversal therefore works better at that age. Also, they're not the real model of Rett syndrome. This is the real model of Rett syndrome, and these animals are no longer young and plastic, so we want to do this experiment. Inject with tamoxifen when the animals are 6 months old or so. And then this just shows that this also works. That's a reversed animal. That's a wild-type animal, a normal animal, and you can see they're indistinguishable. And this is an animal that was unable to respond to tamoxifen because we genetically made it that way. So it still is obese, which is a characteristic of the females on this genetic background, immobile, and it does the hind limb clasping and breathing arrhythmia and all that sort of stuff. So you can, this mouse looked like that mouse when the treatment started. So the implications of this reversibility is that obviously Rett syndrome is potentially a curable condition. And you have to use the potentially word there because these are mice, not humans, but nevertheless it's encouraging. It also means Rett syndrome, like most of the autism diseases, are being called neurodevelopmental disorders. And the implication is that something goes wrong during development in terms, and that you can never recover from that. And I think when one thinks of brain diseases, brain disorders, one tends to think of them as irrevocable. Actually, the experimental evidence to support that is not strong. And this questions that, and there's work with Fragile X syndrome as well, and other so-called neurodevelopmental disorders that suggest that actually they're not neurodevelopmental at all. And in fact, if you take away MECP2 in adults, adults die, certainly not only required during development. And so this reversibility may be more widespreadly true than was previously thought. And that can only be good in terms of exploring therapeutic options. Everything we've done suggests that actually what MECP2 does is sustain neuronal function. These are cells that are never going to divide. They take ages deciding who they're going to be connected to, and in an elaborate dance of synaptogenesis and culling of excess neurons. Then they never get to refresh themselves, and so maintenance is probably a vitally important function. And I think MECP2 may be one of the proteins that does that. So that's just a hypothesis at this point. So prospects for therapy, you could do all sorts of things. And for time reasons, I'm not going to go through this. I'm just going to talk about our attempts to do gene therapy. The dose of this protein is critical. So gene therapy doesn't sound very promising. But I'm just going to show you some results because I think gene therapy, or more likely gene editing, is the logical endpoint of the genomics revolution. Having found all this genetic variability associated with disease, what better than to be able to fix it? And I feel that this is what's going to take a while of basically engineering to find out exactly how we should do that. So this, by comparison with that aspiration, is very primitive. This is an adeno-associated virus. And the experiments here were done in collaboration with Gail Mandel of Howard Hughes Medical Institute in Oregon, and her laboratory, and in our lab, Hélène Cheval did the experiments as well. So we take this MECP2 promoter, we drive a truncated MECP2 gene, not much fits into these viruses, they don't replicate. You then put them in, and there are two ways, and you can go directly into the brain through 6 boreholes in the brain, which is very laborious, and this doesn't actually work very well. But what Gail Mandel's lab did was to use this virus in an unexpected way, namely to put it in the systemic circulation system. So put it in through the facial vein or through the tail vein, and then in females that are 7 to 12 months old, so this is the real Rett model, and ask what happens. And the improvements are quite dramatic. This is the rotor rod. It's a wheel. You put a mouse on, turn it round slowly, and they fall off. But it takes them a while to fall off, and actually the first day they fall off more quickly than the second day when they've learned a bit, and then the third day they're a bit better. The animals without MECP2, or with stopped MECP2 are quite, without MECP2 are quite bad at this, but then if you put in this virus, you're getting a significant improvement. This is the so-called inverted grid test, which is simply taking the lid off the cage and turning it upside down, so it's rather a fancy name for that, and you see how long before they fall off. You see that the red bar is the way they are when you without MECP2, and these are the rescued mice. They've improved a lot. And the third test, and there are other tests I could show you, is the nesting test. You weigh a certain amount of nesting material, you plonk it in the cage, and then the next day you come back and see how much of it they've used to make a nest. So it gives you a number. And not much of it has gone with the mutant mice. A lot has gone with the normal mice, and the rescued mice are vastly better. So I'll finish up by showing you, as the final thing, a couple of mice. This is not as good as an experiment as I showed you before. There I showed you one mouse, before and after. This is, for obvious reasons, a mouse that's had the virus treated with an empty virus. So it has the virus, but they didn't have anything in it. This is the way the symptoms look in the females. That almost you can imagine the limb clasping, very immobile and clearly not that healthy. And then this is an example of a mouse that was like that, but now as a result, it's a different mouse. As a result of receiving the virus, it's vastly improved. This is the data of Sohrab Garg in the Mandel laboratory, with whom we are collaborating. So I would hesitate to say that this is a therapy that necessarily can be adapted to humans rapidly, because the viral load would be colossal. The amount that gets into the brain is relatively small, and 10% of humans have antibodies against these viruses anyway. But it's a basis. So research into the causes of Rett syndrome is currently a hot area in biomedical science. Epigenetics, yes, the epigenome is disturbed in these mice, and brain, autism. These are fascinating areas that are combined in this field. Findings over the past decade has actually changed our perception of this disorder and by implication of others. And by that I mean reversibility, because it was thought to be impossible. The search for potential therapies is going on apace. I showed you what we're doing. We are far from alone. There's lots going on. And no approach is yet proven to work clinically. There is a way to go yet, unfortunately. It's frustrating that one feels one is quite close. If only one could engineer something that would do it. And the goal remains to discover robust treatments that either reduce or even eliminate the burden of Rett syndrome. So just to summarize then, the whole thing, we've ranged over quite a lot. Epigenetics is more or less how the genome of living things is organized and managed. It's a high-level word. Let's not worry about exactly what it means. Every definition encompasses fascinating biology. CG is a genome signaling module. It's very short. It sounds too short to be useful, but I hope you're persuaded that actually it is used as a way of adapting regions of the genome to their function. Proteins that read different chemical forms of CG, unmethylated, methylated, lead to contrasting biological outcomes. And mismanaged, disorganized epigenomes are involved in disease. And the extent to which they're involved in disease is actually profoundly unknown. And for that reason, epigenome manipulation, for example, pharmacologically, may have therapeutic value in diverse human disorders. Epigenetic drugs, so-called, are already in the clinic, histone deacetylases, inhibitors, etc. And there are far more in the pipeline. I would say that we don't really know what they're going to be good for, because there could be all sorts of disorders where global epigenome manipulation has a consequence. And so I think we're in for exciting times. This is my lab. This is not my whole lab. This is just people who were in my lab, whose data I've shown, people who are in my lab, some of whose data I've shown. Gail Mandel, who I'm grateful to for allowing me to use her data, the data that was generated in her lab on the gene therapy. Brian Casper, who made the viruses for her and us. And Stuart and Coleman colleagues from Glasgow University, who are tell us about neuroscience, of which we're ignorant. And finally, all this plethora of funding agencies, from the very small to the very large, who have made our work possible over the years. And this is my lab. I wanted to go to the north of Scotland for our retreat. They wanted to go somewhere hot, and they won. So this is Barcelona again. Thank you very much.
Thank you, Adrian, for a splendid lecture. And Adrian will now take questions for 10 minutes or so. This one over there. If you have a question, if you'd like to ask one, put your hand up in advance. There are 2 roving mics, and we can send one near you so that we can keep the questions coming.
Hi there. You recently published an article in The New Scientist about epigenetics. I wonder if you've got time just to mention a little bit about the post-mortem suicide data that you came across. You weren't able to talk about it at length in your article, but I wondered if you could briefly mention a point about that.
It's not my data. It's a report that people who had committed suicide claiming to have been abused as children had a different degree of methylation of a stress hormone receptor promoter in their brains. And I don't feel, and I'm not sure that the authors would feel, despite the elevated status of the publication, that they have achieved statistical significance with just 12, and also leaving out, as it says in the methods section, outliers that presumably didn't fall within their average. So I feel if the way you treat your children becomes hardwired into their lives through this epigenomic mechanism, I feel before one announces that to the world, one has to be pretty sure, and I don't feel in this case they could be.
At the back.
If there's a guardian of the genome, is there a guardian of the epigenome?
Sorry, I missed the very first phrase.
There is a guardian of the genome. Is there a guardian of the epigenome?
Is there a guardian of the epigenome? I don't know. This is a phrase. Guardians of the genome are proteins like p53 that take steps when the genome is damaged. It's not really clear if damaging the epigenome is quite in a state of flux. There is no one epigenome. That's really the problem with genomic analysis, that every cell type will have a different one. And actually, so it depends on your stance. Some people believe that the epigenome is somewhat fixed and the genes operate within this rather inflexible set of rules. I, from what I have seen, find the epigenome does what it's told by the genes and their transcription factors. In a way, the epigenome adapts the genome to its function as determined by other proteins. Now, there's a question of degree between those two extremes. But I don't see the epigenome as something that gets fixed and then is transmitted forever and you can't do anything about it, and even your grandchildren can't do anything about it 2 generations later. That there may be some of that going on, but I feel it's likely to be far less than is sometimes suggested. Let's dried up the questions.
Another one, 4 rows from the back there. Oh, there's one here.
First, why does it take so long for clinical symptoms to become manifest, both in the mice and in the humans?
Well, that's a very good question. We actually haven't the faintest idea. I mean, you could argue, so in the case of humans, the time when they get it, 18 months of age, is a time of a great activity in the brain. And so that's sort of fueled the idea that this is a neurodevelopmental disorder, and you only really start getting the problems when the brain is going through particular types of dance of the neurons, in particular synaptogenesis. But actually in the mouse, if one looks at that, these mice are 6 months old. They're not going through any developmental processes at all, as far as we know. They're just gradually aging like the rest of us. So in that case, it doesn't quite fit. And I think that the alternative hypothesis is that without MECP2, the functional half-life of your neurons, not their lives, but their functional half-life, is reduced. And you cross some threshold at which the brain stops working properly. But actually, I don't have a satisfactory answer for your question. So it's one of the key questions.
Hi. My question's about gene therapy. And one of the limitations of this is that you can't cause large enough change throughout the entire tissue to correct the fault. And I was wondering whether or not you thought that gene therapy had greater therapeutic implications for epigenetic disease rather than genetic disease.
Well, I wasn't quite sure about the link between the first bit of your... I mean, I agree with the reservation that you have to hit a high percentage of cells. Yeah. Actually, these adeno-associated viruses do that. They've selected serotypes, and they're all naturally occurring. None of them have been engineered in any way, and so there's scope to improve them. But they spread throughout the brain. So the systemic injection, if you look in the brain, it's everywhere. So it does get everywhere, and if you do the injection into the brain, and there are clinical trials now for Batten disease, a lysosomal storage disease, where there are 6 boreholes, and they're putting it in, and they're getting big spread through the brain of this virus. They don't divide, and they keep churning out the protein for a very long time. So I think that sort of thing can be solved. I wouldn't say that gene therapy, this is a blunderbuss approach. You're shoving in an uncontrolled number of genomes into cells, different numbers into different cells. It's the primitive end of what hopefully ultimately will be a rather sophisticated therapy. But I wouldn't say it's for epigenetic rather than genetic. I haven't seen anything that suggests to me that it prefers one or the other.
Well, I was also going to ask whether or not it was the case of being the opposite when you said that the epigenome was constantly in flux. So does that mean using gene therapy to try and correct the epigenome was actually more difficult than gene therapy?
What you've got to remember is Rett syndrome is a genetic disorder. It affects the epigenome. There aren't epigenetic disorders and genetic disorders necessarily. The mutation is a standard mutation in a gene. And it's inherited in a Mendelian manner, if you know, in those very rare families where it's transmitted. So it's a genetic disorder that affects the epigenome. It's not an epigenetic or a genetic disorder.
So Adrian, do the virus-treated mice, do they stay cured? Or do they get ill again when they get much older?
That's a good question, and we don't know, because actually those pictures were only taken within the last 4 weeks. They've lived for 4 weeks, beyond that. But actually, the model experiments say that these things are expressed for a very long time. They don't ever get integrated, and for that reason, it seems they don't seem so susceptible to being shut down by the epigenetic mechanisms that are scouting for strange things in the genome.
Hello. What's your feeling about the big psychiatric disorders like schizophrenia and bipolar affective disorder and so on? Do you think that your research and these epigenetic approaches are going to be important for those?
I don't know. People argue about autism as to whether or not it's more environmental than genetic, and there's now really quite strong evidence that it's genetic, but it's not one genetic disorder, it's hundreds of genetic disorders, actually, literally. There are very many genes that are contributing to autism. That's why it's been so difficult to pin down genetically. So I would say autism, this work is related to, relevant to. It's difficult to know with psychiatric disorders. They tend to, autism, despite being caused by large numbers of different genes, where almost no 2 patients have the same set of genetic lesions, nevertheless have common presentations, features that are in common among their... So it's almost as though when there are problems with the brain, it gravitates towards certain types of behavior. So either it doesn't work at all, in which case survival is in question, or it gravitates to certain types of presentation. So in other words, it's just more about the way in which the brain can cope than about the function of the proteins. Now, I would say schizophrenia, it's a very interesting question. There have, in meetings about schizophrenia and bipolar, always been groups that go off and talk about the possibility that it's caused purely by epigenetics and the environment. But I think increasingly, as more and more sequencing gets done, my bias would be that we will find genetic causes for these disorders. And then all of the panoply of ways of dealing with genetic disorders will be drawn upon to try and fix that. To me, one surprising thing about, if I may just come, the brain, you would have thought, is the most inaccessible place to ever do stuff like gene therapy. But actually, it's quite a good place because the cells don't divide. There is immune response in there, but it's nowhere near as virulent as it is in other places. So you can do more stuff in the brain, and things will spread through the brain. So maybe that will apply to the disorders you're talking about, but it's really a long way away, I think.
In the blue card.
Is anyone working on Friedreich's ataxia?
Friedreich's ataxia?
Yes, that's where the
Lots of people are working on Friedreich's ataxia, but you're not talking to the right person to ask about it, unfortunately. I mean, I would need a quick reminder about exactly what the lesion is there. Is it DNA repair? I can't remember. Can anybody remember? DNA repair, yes. That would be very different. If the lesion's due to DNA damage, then that's not really quite in the league of epigenetics as we've been discussing it. But rest assured, Friedreich's ataxia is a very active area of research.
Okay, there's one there, then one in the green cardigan, and then one against the wall, and we might have to wrap it up at that point.
Fantastic talk, Adrian. It was interesting that you observed obesity in the mice. Were any aspects of feeding or weight changed with the adenovirus treatment?
Unfortunately, we've never really investigated that. I think they don't eat more. I think it's probably, but we don't really know. All I can tell you is when you do the reversal, it's the nicest thing to watch. You just simply watch them come down. Over a period of 3 weeks, their weight goes from being obese to being normal. So, but on the other hand, we're activating MECP2 in every cell in the body. And that actually is an area we're interested in now because are there also peripheral, neuroscientists call everything that isn't the brain the periphery for some reason, but are there peripheral phenotypes that we've been missing perhaps?
Hello, I would just to ask about that. So can you see mosaic cells in other organisms or organs? And does they have
We've never looked.
Some genotype? And another more thing. So if it's not like that, it means that in some cases some cells will be dying because they don't express it. And so they are kind of selected. And because in the brain they cannot...
No, but that doesn't happen, you see. You can, the cells without it don't die. That's why reversal is such an interesting possibility. You know? And in fact, with respect to mosaicism in other tissues, there's quite a variation in the severity of Rett syndrome. And quite often it's due to skewed X chromosome inactivation in the population. There's quite a variation. It isn't always 50-50, half of the paternal, half the maternal. Quite often it's very skewed, you know, like 1 in 10, 1 or 2 in 10. It's very skewed. When the skewing is against the mutated version, the symptoms are much milder. And so you get what's called a speech-preserved version, where you know, there's a spectrum to normality. Now, you measure that by looking at the blood. So whatever you find in the blood looks as though, quite often, it reflects what's going on in the brain.
Okay, thanks.
The one last one.
Thank you. Do all animals have epigenomes?
Yes.
Or is it just a characteristic of the higher animals?
Even yeast has marks on its genome. Actually, yeast doesn't have DNA methylation. C. elegans, this worm, doesn't have DNA methylation. Drosophila melanogaster has virtually no DNA methylation. So the models that are quite often worked on don't. They're chosen, actually, quite often because they have a small genome, rapid generation time. But those animals nevertheless have what one would call an epigenome because they have histone tails covered in marks. So these are quite conserved throughout all organisms that one would call eukaryotes. That's animals, plants, fungi.
Okay, on that definitive note, I think we could have kept on going for much longer with the questions, but Adrian has another rest of his program to get through, so I was asked to bring this to a halt before now, actually. I think you've done rather well out of him. As expected, Adrian has given us a wonderful lecture, very sort of wide-ranging, quite challenging, and he's covered a very large area. So thank you very much for that. And now I'm going to ask Patrick Valence, who is the president for pharmaceutical R&D at GSK, to come and present Adrian with his medal. Patrick joined GSK as head of drug discovery, I think, in 2006, and he's now president of R&D Pharmaceuticals. Is that right? Oh, yes. He's the man with the money. He's actually giving the check.
The check, yeah. They're giving the medal. Adrian, thank you very much. That was absolutely terrific. And it's a real privilege for GSK to be able to fund this lecture and prize. And it's been a privilege for GSK or its precursor companies for 32 years. As Jean said, that we started with the Wellcome Foundation in 1980 and then Glaxo Wellcome and now GSK. And some things have stayed constant over those time and some things have changed. The company is much, much bigger. The company is totally global. And, of course, the whole nature of drug discovery and development has changed. But some things have stayed constant. And one of the things that stayed constant is our base in the UK and our commitment to the UK. And just about 50% of our R&D activity is in the UK. And one of the reasons that we're here is the excellence of the science base. And I think that's been admirably demonstrated today, Adrian, by what you've said, and of course is embedded in the values of this institution. The second thing that stayed constant, perhaps not constant, but in a way started at the beginning and is very, very important to us now, is a very close working relationship with academics. I think it was absolutely the hallmark of the Wellcome Foundation, and I think it's absolutely the hallmark of what we're doing now. And it's perhaps no surprise that I think it was 5 or 6 years ago when we decided we needed to understand what the opportunities were in epigenetics for drug discovery. We reached out to the very leading academics in this field to find out what we should be thinking about. And Adrian, it was you that came in to talk to some of our team to help us get started on that. So it's terrific to hear this today, unbelievably impressive. And Jean gives the lasting thing, which is the medal, and I give the transient thing, which is the money. But I hope it brings some pleasure. And thank you very much indeed. Thank you.